Well, welcome back, everybody. Today we're going to be talking about cover crops and nematodes. For those who don't know, I'm Stetson Maldonado, and I'm here with Tom Johnson.
Hello.
So here's a roadmap about how the talk is going to go today. Today we're talking about what are nematodes, types of nematodes, virus vectors, so how nematodes can spread viruses, and sampling for nematodes. And then I'll toss it over to Tom, and he'll talk about the more practical portions of this as far as crop damage, problems that nematodes can cause, different plant nematode interactions, and then he'll discuss some of the recent research in cover crops as far as nematode interactions. To start off, what are nematodes in the first place? So nematodes are an unsegmented roundworm, and typically they're microscopic. Their name is comes from some Greek words, the first one being nema, which is thread, and toid, which is form, which I think is a good example of just how tiny these things are. They're very much like itty bitty threads. Often, I'd say for the vast majority, especially the ones that we deal with in agriculture, you're going to need a microscope to see these things. They can range from 0.08 millimeters up to 7 meters long. And if you're wondering, how did I just say that we need a microscope for something that can be 7 meters long? This is a very rare case, and I believe specifically these ones are the intestinal parasites of some whales. So they have their own niche. The vast majority are going to be in that millimeter range. And they can eat a huge variety of things, ranging from fungi, bacteria, other nematodes, insect, algae, plants, and animals. I was listening to a talk once about nematodes, and the person was saying that if you removed everything from existence on the planet except for nematodes, it'll still look like we were here because their nematodes are so ubiquitous. They're just everywhere and they're in everything, which is not the warming thought I was wanting to have. Here we have a very basic outline of their life cycle. So nematodes lay eggs. Those can be deposited on the soil or in the soil and or retained in the mother's body. So with soybean cyst nematodes, the females, they'll be buried into that root. They will lay some of the eggs on the outside of that root into the soil, and some eggs they'll actually retain inside of their body. And when she passes away, those eggs can hatch, I believe, over the next couple 8 years or up to 8 years. So there's definitely quite a bit of staying power for some of these. Not all nematodes do this, but I just thought that was an interesting example of how diverse their egg-laying systems can be. Juveniles will molt their cuticles as they grow, so kind of like an insect. Typically it'll be the second molt nematode that would be the one that leaves the egg. A lot of them will molt inside of the egg. And for some species, these are the only ones that are free living and mobile throughout the soil. And we'll learn a little bit about the differences there. And then, of course, you have your adults. And unlike many insects, any life stage nematode can overwinter. So there's different types of nematodes. I probably should have said lifestyles for nematodes. So there's endoparasitic. So endo means in, and then parasite. Everybody's familiar with that. These are parasites that will live inside of the host. So there's sedentary parasites, which would be your root knot nematode and your citrus nematode. These are guys that will settle inside of the plant and then they'll just stop and stay where they are. So in the picture over here, these are I want to say they're a cyst nematode, and those yellow bumps are actually female nematodes, and they're lodged inside of that root, and that's where they'll spend the rest of their lives, right there in that root. There's also migratory endoparasites, such as lesion, lance, burrowing, and foliar nematodes. These are guys that will enter the host and then they'll continue to move around inside of that host. A common issue with these is they'll block up the xylem or the phloem of the plant and kind of throttle that system, almost like a plaque buildup in your arteries. Then there's the ectoparasites. These are guys that are living outside of the host. They'll be going along the edge of these roots and they'll poke holes and travel along as they eat. Examples of these would be sting, stubby root, spiral, and ring nematodes. And then this last one, the free-living nematodes. These are going to be guys that are living throughout the soil. They could be predatory nematodes. They could be fungal-eating nematodes. These will be your guys that are not tied to a host. Tom, was there anything you wanted to add on this slide?
No, actually, the free livings a lot of times are the ones that would be keeping the other ones kind of in check. But they're how they tell these things apart, besides plant symptoms, a lot of times really boils down to what their mouth parts look like, which has to be done under a very powerful microscope. So they have different ways of basically sucking sap out of the plant. The free living ones tend to be closer mouth parts to the ectoparasite with a lancet or a way to grab something else, basically. So they're not the bad ones aren't alone down there. There's other things chasing them around. So that's always handy to know.
Yeah. They're like plants or insects. There's many of them in the same area, but you're only worried about a few. For feeding damage, so the vast majority of plant parasitic nematodes feed on root cells, though some can live on other portions of the plant. Here in this photo, I have root knot nematode, and you can see that it's created all these lumps and bumps and just disgusting looking nodules all over the root. Some can feed selectively on a part of the plant, so some can focus only on the root or only on the cells of the root. There's others that alter the plant growth to create better living conditions for themselves. So this photo is a great example where the nematodes actually released a compound that got the plant to grow these bumps or these capsules to create better living conditions for themselves. And there's others that inject metabolites to break down the cell or break down or kill plant cells. So I like to think of these a lot like spiders. So if you think about how a spider will catch an insect and inject it, and that unfortunate insect will break down and become a slushy, for the spider, it happens a lot like with these nematodes. So I'd like to compare those to the feeding selectively ones. Those are more like a straw. They'll stick a straw in that plant cell and suck it clean. These other ones will actually work to break down that plant cell. So nematodes can also spread viruses. Virus acquisition typically occurs through feeding on an infected cell. And then an infected adult or juvenile will take that and spread it to a new plant with a new feeding wound. So I would say this is primarily coming from your ectoparasites, though I would think that juveniles of endoparasites could also spread these around, depending on the species. Virus transmission by nematodes is typical. Oh yeah, I just said that. Um, specificities. Some nematodes will only spread certain viruses. There's a bit of a relationship between the two. So this is mostly determined by an inherited trait. So there are some nematodes that have, I guess you could kind of describe them as a lock and key system. It just happens this way. But the esophagus will attach to the virus coat, which is the outside of the virus. If you think about that classic virus picture where it's got all those little bumps and things that are sticking out, depending on the shape of those protrusions, it can stick and adhere to the nematode's esophagus, and that way it can ensure that it's carried along with the nematode to its next host or next victim. So for sampling for nematodes, Tom was awesome and went and took this picture. So this is a what do you call this?
That's a soil probe.
A
There's
soil probe?
Oakfield, several other ones. They'll take a sample anywhere from 3/4 to an inch wide for a foot to 18 inches at a whack type of thing. So they're pushed into the soil. They pull it up. You have a circular core now that you would basically scrape off into your container. We'll get into this a little bit later, but for annuals or annual systems where you're starting to suspect you have a nematode issue, you're sampling pre-plant 6 to 8 inches deep. For perennials, anywhere from 4 to 24 inches deep, because that's where the majority of your roots are of your cash crop. And you're going to sample within that root zone, basically. So, and you want to do it kind of in a zigzag pattern. If there's a particular patch that's really bad, you take a separate sampling out of that area, and you would combine soil cores, or you can actually do it with a shovel if you had to, so that you basically get a as uniform as possible sample out of the area you're going at. So, and you mix all those samples basically together and bag them up. For patches where you think there's a problem, like I said, you would take a separate set of samples. Your highest nematode densities are going to be at or just before harvest in an annual crop, which is, like I said, basically you're going to be doing a pre-plant of an annual system. And their life cycle, basically, they like warm, wet soil, so they're going to grow all summer long in your perennial crop. So that would be, you're going to sample for nematodes probably just after harvest or just at harvest, somewhere in there, in that point before the soil starts to cool off. And you can also dig up plants and take root samples out of annuals. And if you happen to get some roots or dig down and get roots out of your perennial crop, basically, you can also submit those.
Yeah. And then I just also wanted to emphasize, if you have a patch, there's another picture further down that's a good example. But if you have a patch of yellow or stunted plants, you want to take those samples from the edge of that patch, not from within the center, just because that's where your highest or your healthiest nematode population will be. They want to be eating your healthy host plants, not your sick and dying ones.
Yeah, and it's not like they went swimming out to the edge of the yellow patch. It's just because there's healthy roots out there for them to feed on now.
Yeah. So in the lab, this is how some of the samples are processed. So for soil samples, they'll be washed through several sieves. So they're trying to wash the nematodes out of the soil. So you can imagine how difficult it is to selectively clean out a microscopic organism from some dirt. There's a there's some appreciation for what they do. Then they centrifuge that to try to compact it, so they have a device that spins. And I think the time of spinning that they'll focus on depends on the lab, but they'll have these little vials, and they're trying to get all of those nematodes to the bottom of that vial to make sure that they're all in one spot. They'll then recentrifuge that with a sugar suspension. Then they'll wash once again through a fine sieve and continue to wash that to remove the sugar, and then a poor victim has to go and count these nematodes with a microscope. So they'll have some slides that have the tiniest microscopic grid that you've ever seen. You can't even see it with the naked eye. And they'll use that grid to try to figure out how many nematodes are in that sample. For roots, it's a little bit easier. So they'll place the roots in a coffee filter, and then they'll place that in a mist chamber for about 72 hours, and then they'll count once again with that same microscope. What I was told as far as the theory about this system is the nematodes they have an aversion to light. So they're going to try to move what they think is deeper into the soil, but they're actually moving from those roots through that coffee filter, and then they drop into the bottom of that container. And that's where they can be counted with that microscope. The results, I want to emphasize, the results are only as good as the samples. So if you took your sample and you're focusing, say, in the orchard drive row, and you avoided all of your roots, and it comes back and says, like, oh, there's very few nematodes, you might have actually missed the bulk of your population, which are over in the roots of your cash crop. So make sure you take good samples. This is a really important slide about how to wreck your samples. So even if you took the best samples, you got the greatest distribution, or the best representative samples, you can ruin your results by, for instance, letting the soil dry before bagging. So these are living organisms. You got to remember that it's alive. You're looking for something that's living. So if you let it dry, then they can die through lack of water. If you aren't very careful with packing these bags before you ship them, they can get jostled around and open up and spill their contents everywhere. So you want to make sure that they're secured. You also, if you're taking multiple samples, you want to label the bags. You don't want to send a box of unlabeled bags to the lab and then hope that they can use some magic to figure out what sample goes for what block. So always label them and secure those bags. Don't just hope that that Ziploc bag is going to stay closed during shipping. So you want to tape that shut. Some people can get confused and send leaf cuttings instead of soil or roots. So um, if you since most of the plant parasitic nematodes are in your soil or in your roots, you want to focus on sending samples from where your pest is going to be located. You want to collect more, at least, or more than a pint of soil, so they have enough soil to run those lab tests on. You don't want to sample dead plants. Dead plants can't support any living organism, can't support living nematodes. So they're either going to have already been long passed away or they're not going to be there. So you want to sample from living plants. Don't expose that sample to heat. So don't put it on your dashboard and then let it bake all throughout the day. You want to keep it protected. So Tom here has a great picture with a cooler. So keep those samples from overheating in the cooler if you, or at least keep them out of the sun. You don't want to send a sample without payment or without a plan for payment. So these people, they do have to pay their own bills, so they're not going to be processing a sample without an idea that they're going to be paid for it. And don't send a sample in on a Friday or just before a holiday. People are just like us, and they don't want to be working on a holiday or on the weekend. So if you send something and it arrives there when nobody's there to collect your sample, it can end up baking on the in the mailbox. Or you wanting to say something, Tom?
Oh, yeah, or it just sits in front of the door until they come back on Tuesday after the 3-day weekend, and it was
Mm-hmm.
105. So that was like, yeah, that sample's probably not good anymore.
Cooked dirt. And then using results. So each of these are going to be different based on the lab that you use. So the action threshold is the population level at which prevention is required to avoid economic damage. So since nematodes are everywhere and they're going to exist in your soil no matter what, just because you have a nematode doesn't mean that you need to treat. But if you do have an increased population or a population at damaging levels, then you do want to treat. And you'll often get a recommendation from the lab. This can vary by your state, by your crop, your soil type. It can vary by the local research and from the lab's own personal experience. So when you're using a lab, you want to stick with the same lab time and time again, just because the sampling techniques and the reporting techniques can vary quite a bit. As far as risk levels, so this isn't about the population, this is about the type of nematode. So some nematodes can have low, moderate, or high risk, and this will also vary by those same factors, state, crop, soil type, and so forth, and also by lab.
And I'd like to interject just a little bit here is something we kind of missed is if you're going to sample for nematodes, expect to do that on kind of a regular basis. In other words, you don't in a perennial crop, you probably don't have to do it every year unless the first one came back and says, yeah, you got lots of them, which is your action threshold. Then you have to determine what you potentially are going to do about it. And then if you did do something for it, you want to sample again next year about the same time to see if you made a difference or not. So yeah, you want to, just like soil fertility sampling, you want to do that on a schedule, whether it's annual or biannual or every 6 months or whatever you want to do. Just make sure you continue to follow through with that.
Yeah, thank you for pointing that out. I'd forgotten to mention, like with orchards or vineyards, you definitely want to be sampling pre-plant and treating pre-plant, and the options available to you will be different depending on whether or not your crop is there. So here we have crop damage, and I'm going to toss it over to Tom.
So, okay, crop damage. is going to, of course, depend on whether you're talking a perennial crop or an annual crop system, and then what nematodes you may be up against. But the majority of the time, mentioned everywhere you look for it, the above ground symptoms tend to be stunted growth, smaller leaves, poor nutrient uptake, which is a lot of times symptomatic of poor nutrition of some kind or another. Lower yield, sometimes that's hard to determine until you've done something to reduce the nematode load that you might have. A lot of times what will show up is premature wilting during dry periods or when the irrigation scheduling gets a little bit long out there. That kind of goes with the smaller leaves and the stunted growth. Or you may get a thinning of stand, um, depending on the nematode, depending on the crop, as usual. There are some that will, yeah, I just knocked it out and killed it. And your stand's thinner than you expect. In this situation, you've got those plants are all kind of stunted, probably due to some soil-borne infection. Of course, these above ground symptoms are all similar to drought, stress, or other diseases. So you're going to be doing a little bit of forensic agronomy out there, digging up some plants and looking at what's going on in the root system, because that's the nematodes clog up the root system. That's why the plant can't pull up water or nutrients and basically looks stunted and unhappy. So if you did go out there and start to dig up plants and you're going to be looking at roots, your below ground symptoms a lot of times are going to be galling on the roots, picture earlier. Knots, which is slightly different than galling. The knots are the larger lumps, basically. Don't confuse either of these two, basically, with nodulation by rhizobium in legumes, which can be kind of hard to figure out until you cut it. Legumes, if you cut the nodule in half, hopefully it's red in the middle. In a nematode issue, it's probably going to be black or not discolored. You're going to may see some root dieback, especially in the tips. Occasionally you'll see lesions. You might have fewer roots. A lot of times what you'll see is when you've had that root tip dieback, you'll have secondary infections of soil-borne pathogens, bacterial infections or fungal infections, because that root tissue's been compromised and that has a way for it to get in. So a lot of our common problematic nematodes, and when this is not, this is kind of a list of villains, but it's not necessarily in order. It's of course going to depend on your crop. So the dagger, root knot, pin, cyst, lesion, those are kind of the big heavy hitters that we run into in crop production that will damage the roots and cause you to have yield drag or whatever type of thing. You kind of want to know what you suspect when you're ordering lab samples. So part of that goes back to that diagnosing what's going on out there. You're seeing galling or you're seeing knots out there on your roots. Well, then you're probably looking for either a dagger or a root knot nematode, maybe a pin. Cyst tends to be, if I remember right, on the outside, and it looks like little white eggs laid on the roots, on the outsides of them. Lesion's the tough one because it moves inside the cell, inside the roots, and then moves around a little bit. A lot of times what you'll find in that is root tip dieback, or you may see a little bit of discoloration in feeder roots. But a lot of times a general sample goes in. Sometimes it'll be diagnosed, and sometimes it's like, yeah, if you don't tell them what to look for, they're just going to tell you you got X number of nematodes, not necessarily dividing them out again, because again, identifying them beyond just saying, yeah, you have X number in a sample is takes a little bit more work. Takes a much stronger microscope a lot of times. So like a lot of other things in plant diseases as well, because we're dealing with stuff in the soil, you may be looking at non-crop host plants that might be causing you a problem. They might be carrying on that, letting that nematode population continue or even expand to some situation. So a lot of times it's the same family as your crop, same plant family as your crop, or similar root system, root zone as your crop. That type of thing's going on out there. So your plants, which could be those nematodes are feeding on, or used to complete their life cycle, but aren't a crop. So you kind of want to check which species in your areas can harbor your suspected pest nematode, just like various other pathogens in the soil. And then if you can, you want to rotate, if you're an annual system, you want to rotate into other things that won't necessarily exasperate your problem. So one of the techniques in trying to keep nematode populations in check is a rotation with non-host plants. So a non-host plant is basically a plant that either the problematic nematodes can't feed off of, or they can't complete their life cycle within that crop. So they starve, or they don't reproduce as well. And sometimes it can be reduced or kept in check with one year rotation. Sometimes it may take several years of hammering that thing to try and bribe that population down. Some of those examples are going to be things like barley or small grains. Merced rye is very good at it, which is something we use quite a bit. It's a spring rye in California, grows real fast in the winter. So same with barley. Fibrous root systems, they're not going to, root knot's going to have a hard time doing much with that. Sudan grass not only a non-host, but it's kind of antagonistic as well. It exudes chemistries from its roots and its plant tissues and breakdown that tend to like drive stuff off. And then sun hemp is actually a legume, warm season legume, that is considered a non-host. So it's something to consider. It works well in some things and other not so much in other things. I know locally in California, our window of being able to do a cover crop is usually not going to favor something in the warm season. But in other locations, it may actually have something to do with that. And then, of course, there's antagonistic plants. These would be plants that exude those chemistries within the soil or as the plant material breaks down that will interfere a lot of times with the life cycle of the nematodes in the thing. They either produce basically chemistries within the soil atmosphere that are detrimental to the nematodes, or they're just basically they're non-host, or they just you can't reproduce on it, but a lot of times it just actually outright kills them. So some of those examples are marigolds. We mentioned brassicas as well. Brassicas actually can host some nematodes, but on the other hand, the plant material breakdown can cause a certain amount of damage to populations. Phacelia is also a non-host antagonistic plant. And there's some other ones out there as well that exude that allelopathic enzymes against other plants and sometimes against nematodes. They can't be taught using these plants can reduce those populations, but you can't really rely on them for total control. So then we get to our control systems, and this is, again, depending on your crop. If you're replanting, of course, we're going to recommend that you sample for nematode before you put trees or vines back in the ground, type of thing. And the number one thing everybody kind of goes to a lot of times in modern agriculture, of course, is soil fumigation. Some places, especially in California, it's becoming tougher and tougher to do, to have effective materials that will limit or reduce or eliminate nematode issues. Comes with some regulation and all that fun stuff. And you're going to be doing it pre-plant. It's not like you're going to go out and be able to fumigate a standing perennial cash crop. The biofumigation idea is this is where we would use the brassicas, and basically what we want to do is convert that above ground growth into organic matter that's breaking down in the soil atmosphere, and that causes a similar action to fumigation. It only works basically as deep as we can get the biomass worked in and how fast we can make it break down as to how well it works. But in some situations, it works very well. People in the basin in Washington have found it works extremely well for them in eliminating nematodes on their row crops and vegetable crops up there. And It works to some extent in California. The biggest trick usually is getting it handled. Another one they're working right now is anaerobic soil treatment. And this basically is a situation where you put down a whole lot of fairly fast degrading organic matter and you keep it wet and you keep it covered for an extended period of time, and you basically burn the oxygen in the soil, and that kills off the nematodes and a few other fun things out there. It works. It's fairly short term from what the plot trials I've seen, but it works. It would probably work better in annual crops than it would be in perennial crops. Again, differences in root system, differences in how you're going to do things. Solarization using plastic, generally clear, and there are specific films that work very well for this. And it's spread on the soil in the summertime, at least in California, basically, so that we're basically heating the soil up down to about 8-10 inches at the most, type of thing. It helps if you have a way to keep that wet as well, or moist. And again, you're steaming slash heating the soil to solarize or sterilize the soil from at least nematodes, and certain other pathogens are affected by that. One of the longer term ones, but fits very well in perennial crops, is basically soil biology. If you realize that down there in the soil, it's a jungle where there's something that eats something else, getting our soil back to a healthy status where there's a big enough population of just about everything down there is also going to help with your plant problem nematodes, is because there's something else down there that eats that. So you need to work on your soil health and your microbial diversity within the soil atmosphere. It works fairly quickly to start, and then basically needs to be maintained so that you have that action going on for you. And then finally, there's sanitation. Just like for soil-borne disease, just like for a lot of weeds, basically, if you're coming out of a field that you have a known problem in, make sure you're cleaning your equipment before you go into a field that you don't have a problem in. It makes good sense to do that no matter what. But on the other hand, when you're coming out of a heavily infested field and going into another one that probably doesn't have a problem, you really want to make sure you're washing that equipment. You're not carrying inoculant from one place to another. And then, like I said, I am a proponent of cover cropping and soil diversity. And what they have seen, there's some data out there now that they're getting published finally, but there's a lot of anecdotal data out there as well about basically using a cover crop, reintroducing diversity into your perennial system suppresses your plant parasitic nematodes by diversifying the soil biology. You're bringing back more biology in the form of bacteria, fungus, good nematodes, other arthropods running around out there. A lot of the nematodes are actually controlled by fungus that will basically reach out and grab that thing as it swims by and suck it dry. So it's a tiny little war going on down there that we can't hardly see, but keeping everybody fed and keeping everybody at the table is definitely going to help with reducing these nematodes. And some of the suppressive cover crops can affect that as well. I mean, there's, like I said, we talked about antagonistic plants, we talked about non-host plants, using those in a rotation rather than just rotating the same 2 or 3 crops constantly. Putting something different in there for a little while gives the soil a break, helps with the soil biology, but it also can help suppress that because there's nothing for that particular pest nematode to feed on. And then basically, we continue to bring you these things, and we always want to say thank you to our donors. Seeds for Bees, the program that we at Stetson and I operate, basically is entirely funded by the generous donations of individuals and corporations. The neat thing about that is then you can take our program and stack it with a federal or state grant with no interference. We're deeply grateful to the people who make our program possible, many of whom have been with Seeds for Bees since its inception. Almost all of them are great proponents of cover crops and understand kind of what's going on in the soil. So thank you again, and have a good day.
Awesome.